Welcome to Season 2 of Public Health on Call, a podcast from the Johns Hopkins Bloomberg School of Public Health. I'm Joshua Sharfstein, Vice Dean for Public Health Practice and Community Engagement, and a former Secretary of Maryland's Health Department. Our goal is to bring scientific evidence and experience to the public health news of the day through informative interviews with scientists, community leaders, policy experts, public health officials, clinicians, and more. If you have ideas or questions for us to cover, please email us at publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes. Today, Stephanie Desmond talks to Professor Diane Griffin, a Johns Hopkins immunologist, about the vaccines being developed against COVID-19, most notably the RNA vaccines that use a new technology to provide protection from the virus. Let's listen.
Diane Griffin, thanks so much for joining me.
Oh, you're welcome.
I'm excited to have you here today to talk about these RNA vaccines that we've been hearing so much about. So could you tell us a little about what these RNA vaccines are and why they're so different from what we've seen in the past?
Well, they're made of RNA, so that's fairly obvious. But the way they're different, and then also to describe RNA vaccines, is what we want to do with any vaccine is actually deliver a vaccine for a virus, at least, deliver the viral protein that you want to make an immune response to the person that's being vaccinated. And so there are lots of different ways to deliver that and different forms of that viral protein. One is to give the whole virus, inactivate it so it doesn't make the person sick. That's being used by some of these coronavirus vaccines. Another way is to deliver the nucleic acid that actually encodes the protein and let the cell actually make the protein that is going to immunize the person. So that can be done in two ways. There are DNA vaccines, but then they have to make the RNA. So the RNA is the nucleic acid that codes for proteins that cells make. And so basically it has all the information needed to be able to synthesize the protein. So the idea of these vaccines is that you just deliver that RNA that is encoding the viral protein that you're interested in and let the cell actually make that protein and stimulate then the immune system.
What are the benefits of this?
Yeah, so it has several advantages. One is that first of all, because the cell itself is going to make the protein, it's much more likely to be the native protein or to have the right folding and antigenic properties of the protein that the virus would also have. Another big advantage, which is what's come out recently, is that they're fast to make. So if you have the basic platform, and we could talk about the platform in a minute because that does matter, that you're just going to have to insert the sequence for your particular protein of interest. You can just take one out and put another one in. So you have the background of how to make the RNA already there. And so it's very fast to substitute in a new, different coding sequence for a protein. Also, the manufacturing, it turns out to be relatively straightforward for these types of vaccines. That you don't have to grow them in cells, or a lot of things that make manufacturing either difficult or having to go through a lot of hoops with the FDA to prove that your cells are not contaminated with something, et cetera. It sort of bypasses all those steps. And so I think that the fact that they work has been particularly, I think, exciting. It has required a fair amount of sort of manipulation of what the RNA is that you give, because RNA intrinsically is unstable. and so it gets chewed up, and you can't just give RNA. It won't get into cells. The cells won't be able to make the proteins that you want them to do. So these RNA vaccines have been engineered really in order to, first of all, to make them somewhat less likely to be what we call reactogenic or make you sick when you get the vaccine. And that's still somewhat of an issue with these vaccines, is that they're fairly people don't die, but they could be pretty miserable for a while after getting them. But also they have to be put inside of something. And so what's currently being used, and I think it's turned out to be most successful, is a what they call a lipid nanoparticle, which basically surrounds that RNA that's going to be the vaccine and does two things. First of all, it protects it from being degraded. Second of all, it gets it into cells because you can design the nanoparticle surrounding so that it interacts with cells and then goes in. And then the RNA can be released for the cell to actually make the protein that you want.
These are the first RNA vaccines, I understand, that have gotten this far in the process. I guess there was a lot of work done leading up to this moment that made it sort of the perfect moment to try these. Is that right?
That's right. They probably for the last 10 years, people have been working on these kinds of vaccines. And there's two versions. The one that is the current two vaccines that we're dealing with are just the messenger RNA in these particles. There's also a version that's been worked on and maybe for a longer period of time, which is called a self-replicating RNA, where it also has the information needed to copy that RNA. So you make more copies of it, not just the ones you put in, but additional ones. And they've been worked on mostly for cancer vaccines. I think that's where the bulk of the work has been, but it was certainly perfectly adaptable and also has been explored for actually quite a period of time with these sort of prototype, both self-replicating and just non-self-replicating vaccines for immunization purposes as well. So a lot of background information about modifying the RNAs so that they don't make you so sick, and also perfecting this packaging that you need for the RNAs.
So at the moment, these appear to be very successful, the two candidates we've been hearing about.
Yes.
Why were we able to adapt it for the virus that causes COVID when they couldn't really get it into production for these other things they've tried?
Well, they have had prototypes for influenza, for rabies, for a number of other, but they've never gotten to phase 3 testing. They've had earlier stage testing. I think maybe that the need was not so emergent and so critical as it has been here, which is really motivated to move the technology forward fast. But as you say, there's really been a lot of work that's been ongoing for the last 5 to 10 years with this platform.
So what's the potential sort of going forward? I mean, does this mean that we will really be able to react quickly to things? Does this mean perhaps we could create a flu shot that's more effective? I mean, I know flu vaccine is still made in eggs for the most part, which seems just archaic to me.
Right. Yes, it is. Right. And so I think that, yes, I think that we're very likely to see other vaccines. I think the other thing that happened is with any vaccine where we already have a vaccine, where we know it's safe, we know how to make it, usually know how to make it pretty cheaply, then the bar for using some new technology becomes fairly high. You have to want to substitute. You have to have a good reason to want to substitute what you already got. So this came along as a brand new virus. And so it sort of opened the way for really applying these newer methods. And I think that once this turns out to be successful, if it does. I mean, I'm fairly optimistic, but that at least in the short term, it's going to be successful. I think there's still questions about how durable this immunity is, et cetera. But yes, I think it's likely, certainly, to get applied to more emerging infectious diseases. So where we have other new viruses, they're usually viruses coming on the scene, but it may embolden people somewhat to try it for, particularly for new influenza vaccines. And there is work using this platform for influenza vaccines as well.
Are these generating stronger immune responses than traditional vaccines?
Not necessarily. I think that they're good immune responses. I think that but I don't think they're necessarily better. I think we've just been happy that they were good.
Yeah. Now, the one thing that I've been seeing is that one issue with them is that the temperature at which they must be stored. Now, I know that the Pfizer vaccine needs to be stored at like negative 70 degrees Celsius.
Right.
And I saw that Moderna is negative 20 degrees Celsius, which is more like a regular freezer, I guess.
Right.
So
yes.
Do these need to be kept cold because of the RNA?
Yes.
That's why. Okay.
That's why, because the RNA is unstable at higher temperatures. I think that it may well be that the Pfizer vaccine can withstand being a little bit warmer than minus 70, but it hasn't been. It probably just hasn't been proven that. Basically, what the drug companies need to be able to do is to say that we left our vaccine at whatever temperature it is for 2 weeks, and then tested it, and it was just as good as if we didn't. So those kinds of things all take time. And so I think it's probably been easiest just to say, we know it's okay at this really low temperature, so that's what we're going to use.
Although for practical reasons, it feels like some urgency perhaps for Pfizer to find that it can be held not quite that cold.
Yes, I would imagine, I don't have any inside information, that they're working on that. And that's because the Moderna vaccine can be stored at a higher temperature, or a more reasonable, more easily available temperature, that Pfizer will be investigating that possibility for their vaccine as well.
So not only was this developed quickly, it feels like it can be manufactured more quickly.
Yes.
So that's, I guess, another advantage here.
Well, I think that it's as I say, it doesn't require eggs. It doesn't require cells. It just requires, sort of ingredients that you mix together in the manufacturing facility. Now, these vaccines have never been manufactured before, though, at large quantities. And so scaling up is not always as straightforward as just doubling the recipe. So, or 1,000-folding the recipe, which is what you need for this. So I think that that is still an issue. But as you know, the manufacturing facilities sort of anticipated the success of the vaccines, and that I think both companies have invested in manufacturing facilities and even manufacturing the vaccine in advance of getting the data from their trials.
Mm-hmm. You've been doing this a long time. You must be really excited to see a whole new category of vaccine to come out.
Yeah, well, we did some of we work on measles, and there have been reasons to try to develop a new measles vaccine, although it falls into the category of we have a measles vaccine, and so that we know how to make, and it's cheap, et cetera. But we worked on some of the prototype, the self-replicating vaccines for measles, and in monkeys we never brought it to as far as testing it in people, but they work very well. And so so I've been interested in this technology. The self-replicating versions are based on alpha virus replicons, and that's the other virus that we work on, are alpha viruses. So it was a combination of being able to look at both of those. So it is exciting to see the field moved forward was well before we knew that you had to modify the RNA and how you had to package it and that sort of thing. But
Well, Diane Griffin, thank you so much for joining me.
okay, you're welcome.
Public Health on Call is produced by Joshua Sharfstein, Lindsay Smith-Rogers, Stephanie Desmond, and Lamari Morales. Audio production by Spencer Greer, Niall Owen McCusker, Cian Oates, and Matthew Martin, with support from Chip Hickey. Distribution by Nick Moran. Thank you for listening.